So RNA splicing usually begins with a pre-mRNA, which contains a 5' for stability. It contains exons and introns. Exons are known as being expressed, whereas introns have non-coding segments. So in order to make mature mRNA, the segment must be continuously expressed, and so to do that, you must cleave out the intron, which is done through the use of snRNPs, which act as ribozymes, and they come together with other proteins to create a spliceosome. And so the snRNA inside of these snRNPs will then base pair to certain nucleotides in the exon-intron junction, and this will help cleave out the intron. And so the cutout intron will then go to the cell and be degraded, and then the leftover components, the two exons, will then be spliced together to be continuously expressed. And it will also contain a 5' and a poly A tail for stability, and also to prevent degradation in the cell. There's also alternative RNA splicing, where multiple mRNA molecules are created from the same primary transcript. It just depends on which segments are considered exons and which are considered introns. So this is the trip operon, which is usually known as being on because there is an upstream regulatory segment which codes for an inactive repressor protein. This is known as being inactive because it does not fit with the operator. Therefore, RNA polymerase is allowed to transcribe the genes of the operon and create, and this codes for polypeptide subunits of the enzymes that are used to create tryptophan. So, RNA polymerase usually binds to the DNA with the help of a box called the Tata box in the promoter. The Tata box acts as an initiation complex that will help RNA polymerase bind to the DNA. So when there is an abundance of tryptophan, the tryptophan will bind to the inactive repressor, and this will change its shape so that it will become active. So this active repressor will then be allowed to bind to the operator, and this will halt the production of the synthesis of RNA, and this will stop the synthesis of polypeptide subunits of the enzymes that help create tryptophan. So when there is an abundance of tryptophan, usually tryptophan will inhibit the activity of the first enzyme, and this is known as feedback inhibition. It can also repress the expression of the genes of the operon that express all of the subunits for tryptophan. So this is the lac operon, which is usually known as being off because there is a regulatory gene that codes for an active repressor protein, and this the shape of this active repressor protein allows it to bind to the operator. Therefore, RNA polymerase can no longer synthesize RNA. For this to happen, however, for the synthesis of RNA to happen, there is allolactose, which acts as an inducer, which binds to the active repressor protein and makes it inactive by changing its shape so that it can no longer fit with the operator. So this allows RNA polymerase to synthesize RNA. So the genes of the lac operon code for these three enzymes. B-galactosidase is used to hydrolyze lactose into glucose and galactose. Galactose. Permease is a membrane protein which allows glucose inside of the cell, and transacetylase has an unknown function. So this is the enhancer, which consists of three distal control elements, which are known as binding sites, where the activators will bind to them. So next, a DNA bending protein will come and it will bend the DNA over so that the enhancer will be nearby the promoter. So also RNA polymerase, a group of mediator proteins, and general transcription factors will be nearby. So the mediator proteins will then bind to the activation sites of the enhancer, and the transcription factors will bind to RNA polymerase. I'm sorry, will bind to the mediator proteins, and so this will allow RNA polymerase to be on the promoter, and this will enhance or amplify the synthesis of RNA.